everyone, welcome to the 2022 UC Davis Plant Science Symposium. My name is Yufei Qian. I am a Ph.D. student in the Horticulture and Agronomy graduate group, and I'm working with Dr. Gail Taylor on watercress breeding project. I am very happy to show you the inside of the UC Davis indoor vertical farm. My current ongoing research is screening a population of wild accession watercress using the indoor vertically grown settings. To take a closer look of this indoor vertical farm, it is a 40-foot long repurposed shipping container that has a complete hydroponic system without any natural light. All the light sources comes from the LED grow light. Here is a better view of the red versus blue LED lights. To furthermore introduce this growing system, it has a constant supply of nutrient-rich water from the top emitters over every growth panel, and water is recycled in this system, which can save water largely compared to traditional agriculture. Also, at the same time, there are climate sensors. Here are the nutrient tanks and dosage panels, which are inside this facility and can automatically adjust the growth environment. Growers are able to track the environmental parameters and control settings remotely using this online mobile app platform. Now you're seeing an ongoing germination of watercress that I am preparing for a new trial. There is no soil, and I am growing all the seeds on peat moss and cocoa coir plugs. Each seedling gets its own tag with a QR code on it for easier tracking in later stages. At this stage, all the young seedlings are receiving red and blue LED lights as the light source for photosynthesis. Once watercress seedlings develop 3 to 4 true leaves and a formation of healthy root, they're ready to go into the growth wall, which looks like this after transplanting. This leafy green crop contains the most abundant aromatic glucosinolate across the Brassica family. This phytonutrient is a secondary plant metabolite, and its hydrolysis product, isothiocyanates, have been proven to have anti-cancer, anti-inflammatory, and antioxidant effects to human health. From a previous preliminary research, one commercial watercress cultivar was grown following the steps that I just described in 2 different light regimes, 20% versus 30% blue LED light. In this vertical farm, I collected data on plant morphology, yield, and nutritional profile from both treatments, and compared this data set with 2 previous field trials that Lauren Hibbert and I were running in 2019 using the same cultivar. These 2 field trials were in Davis, California and Hampshire, UK. As you can see from the slide, watercress in the UK site was grown within a traditional outdoor gravel bed using stream water, and the US site was an open field with an irrigated soil bed. The growth environments were distinctively different across these 3 trials. Here comes some interesting findings. Yield and glucosinolate quantity were significantly increased in a vertical farm system enriched in blue light relative to field-grown crops. Additionally, PBITC, a glucosinolate not previously identified in field-grown watercress, was only found in plants grown in a vertical farm. The most valued harvestable part of this crop is leaf in the commercial production, and vertical farm-grown watercress had the largest leaf area. Yield increases may be explained by a consistent optimal nutrient and temperature environment that is achievable in a vertical farm. Glucosinolate content tends to increase under short duration exposure to blue LED light, and that may explain the quantitative and qualitative changes of the glucosinolate profile in this vertical farm-grown watercress. If you are interested in reading more about this work, please simply scan the QR code and feel free to reach out to me or Professor Gail Taylor via email, lab website, or our new favorite social media Instagram at TaylorLabUCD. Thank you very much for listening.